Many thanks for choosing us. The Food and Drugs Authority has defended its validation of the test kit currently being used to screen for COVID-19 in passengers arriving in Ghana following the opening of the Kotoka International Airport to commercial flights. It follows concerns that kits may not be as effective in identifying persons with the virus as gold standard PCR tests. There have also been concerns that anything less effective than the PCR test should not cost as much as $150 being charged the passengers. Dr. Kofi Boni is lead virologist at the Noguchi Memorial Institute.
I don't know what they are doing, but I know about between $10 to about $20 for one test. Yeah.
But we are told that it's costing about $150. I don't know if you've heard about it.
Well, maybe so many other things come. If you are buying the kit itself, but maybe I'm sure they are charging administrative and also maybe the personal protective equipment and all that they are using. They will factor it in. Scientifically, the PCR test within 72 hours is enough. I don't know why we have to do an antigen test which is less sensitive upon arrival. If you look at the tests that have been done over the years, we have a varying sensitivity percentage between 34 to around 80%. So if you are looking at these figures, that means about half of the people who will take antigen tests, the results may not be correct. No, about between $10 to about $20 for one test. Yeah.
But we are told that it's costing about $150. I don't know if you've heard about it.
Well, maybe so many other things come. If you are buying the kit itself, but maybe I'm sure they are charging administrative and also maybe the personal protective equipment and all that.
The FDA, in a statement, however, vouched for the efficacy of the test kits. Head of its laboratory service department, Kakari Boateng, explains that the device being used at the airport is not a rapid diagnostic test kit, as has been suggested.
Since March up to date, we have tested this RDT device and this rapid diagnostic test kit. And the sensitivity we told you ranges from 4 to 62. That is why the FDA in its wisdom has never approved any antigen or antibody rapid diagnostic test kit for the detection of the SARS-CoV-2 virus.
Okay.
Yeah, that is clear in our statement. So if you read it, you see.
Yes.
Then you are now talking about the immunofluorescence. Yes, this is the first device that we have approved here, but there are subsequent devices that are still going through the process. And it has also been approved by other stringent regulatory authorities as stated in our statement. And on our website, the guidelines for devices are there. The gold standard is PCR because now there's no international reference antibody or antigens available. So it is compared to a PCR, which is the gold standard now. Okay. And we tell you that specificity, the machine should be specific, that if you have SARS-CoV-2, it should tell you that SARS-CoV-2 and not any other viral disease. Okay. That one we say it should not be less than 99.0%. It's in our release, it's on our website. Then for sensitivity, how accurate can the machine detect if SARS-CoV-2 is present? That one is also 99%. So for a device to be qualified and approved and be put on the market, it has to meet this acceptance criteria.
All right.
And this was what was done.
Mr. Boateng had indicated the FDA only approved the kit because it approved to be highly effective with sensitivity of 99.9%. He also gave the brand of the kit as FER6. Joy News checks from the website of the company shows the manufacturer's sensitivity claim is not up to the FDA's own benchmark. Joining us via Zoom now is an immunologist with the West African Center for Cell Biology of Infectious Pathogens, Dr. Bediako, who has been following the conversation about the test kits in question for a while now. Thank you, Mr. Bediako, and welcome to Joy News Prime.
Thank you for having me.
Dr. Bediako, yes, so you earlier on said you were going to check the kit that is currently being used and check its efficacy. Our checks with the manufacturer, however, indicates a sensitivity below what the FDA specified. Will it be accurate to say it doesn't meet the FDA's own standard?
Well, I mean, I think that question actually has two answers. So yes, I mean, I can see from the screen that the document you are showing is, I believe, is a brochure. It appears that in that document that you indicated to me that the sensitivity, I think, was listed as 87.5. That is in line, I would point out, with sort of what many of us scientists would expect for an antigen kit. As I think in your clip, Dr. Boni said, between 30 to 80% for antigen kits. He was not referring in that case to SARS antigen. He was just saying in general for majority of the antigen kits that are available. And I'll point out that this technology is not new. So it's new. SARS-CoV-2 is a novel virus. So we've been working to develop new kits and new technologies, new ways to detect this virus. But PCR, antigen kits, antibody kits have all been around for a long time. And so for majority of antigen kits, you'd expect close to 80%. So this manufacturer, I believe in this, is claiming that it's around 87.5 sensitivity compared to PCR. So you can see clearly that the manufacturer is saying that this is inferior to some extent to compared to PCR. So this number does appear to contradict to some extent what the Ghana FDA is reporting. Now, I would assume that the Ghana FDA themselves have conducted their own validation studies. So perhaps in their hands, in their validation studies, they got a much higher sensitivity for the device. But typically manufacturers are known to put their best foot forward. After all, they want to sell their device. So it's a little unusual, certainly, for the manufacturer's listed sensitivity to be lower than what the regulatory body has found.
Doc, having verified this, what then would you say are the implications of using this device?
Well, I mean, with any device, any test, there's always a limit of detection. I believe I was on your channel, your program earlier in the day with Mr. Boateng himself. And we both agreed that, limits of detection, every kit has a limit of detection. And so when you are choosing a kit for a particular purpose, you have to ask yourself, what are we going to use that kit for? So basically, a sensitivity of 99% means that 1 out of every 100 positive cases will be undetected. Now, a sensitivity of 87.5 means 13 people or 12 people out of every 100 who are positive will be undetected. And I believe it is not for me really to decide which is acceptable. It is for the Ministry of Health, the Ghana Health Service, to decide what is an acceptable limit. Obviously, PCR tests cost more and take longer. The advantage of these antigen kits is that they are faster and can be deployed in a setting of an airport more easily. But the question then is, do we accept the trade-off for sensitivity? Of course, all these people coming in already have a negative PCR test. So the question then is, maybe the risk is low enough that we do not think that we need a very sensitive test. On the flip side, these people already have a negative PCR test. So if we are to capture anybody who has turned positive between their PCR test and now, we need a very, very sensitive test. So I think these are the questions that need to be more clearly answered. Certainly the discrepancy between the 99 and the 87, that I assume that the FDA probably has some data to provide answers for that. That is not for me to say. The manufacturer says one thing, FDA has their data. It would be nice to see some more agreement between those numbers.
Certainly.
But in terms of accepting a threshold, that is for the Ministry of Health to decide.
We'll seek clarification from Ministry of Health. I'm grateful for your time. Dr. Yalbe Diakon is a immunologist with the West Africa Center for Infectious Pathogens.